Hi and welcome. Avinash Jaiganesh started off listening to his relatives who were pursuing their PhDs and soaked in a lot of scientific temperament. That seems strange, but it's also unusual for a child to be interested in science so early on. But it defined what he was going to do. And today, he's a senior scientist at Ensoma in Boston in the US, and he pursues molecular biology. This is episode 77 of My First Job, the podcast, that covers engaging career journeys, and every single one of them is full of twists, turns, and insights. My name is Venu Gopal Nair. I'm the host of this podcast and CEO of Ideascape Communications, an advertising agency into branding, creative, and design. Settle in, sit back, and enjoy the conversation while I bring Avinash on board. Hi, Avinash, and welcome to the show.
Hi, Mr. Nair. Thank you so much for having me. Very excited to talk to you today.
Wonderful, Avinash. So, like I started off, what exactly got your interest going in science?
Well, my whole family, as you mentioned, is quite scientifically inclined. I would like to say, I got introduced to the concept of scientific pursuits when my uncles and aunts returned for the summers from the US, came to visit family. We spent a lot of time together, and ever since 5 or 6 years old, I would just hang out with them the whole time that they were there. And I would be very curious as to what they were doing, and I was like, "Oh, you're doing research. What's research? You're doing a PhD. What's a PhD?" And so on. And slowly, I started getting a curiosity about the whole process of scientific thinking, the scientific process itself, uh, how to further your knowledge by questioning. It's a big and exciting world of the unknown, right? And that's where the seeds were sown for this curiosity, for
Okay.
finding out about things. And my mom, being a toxicologist, was very much into different biological processes and phenomena that she had researched in her masters and stuff like that. So she would always educate me about biology, the world around me, life and living things. And I got the curiosity to actually question this and say, what is life? What brings about life? What makes our bodies tick? What makes biology happen? So that's kind of how I ended up in my career.
Okay. So, what was school like? Was school distracting because the system of education in India is more rote learning, but you had to you managed to bypass
That,
that.
that makes, I mean, I was good at what I was good at, right? Like I loved math, I loved biology, I loved physics, I
Okay.
loved chemistry, because they helped me understand more about my passions. Didn't do very well in the other subjects, right? I was actually pretty good at language as well. So I like language because of its poetic elegance and so on. But yeah, I just followed my passions. I scored well in where I could apply this, and wherever rote learning was involved, I was quite poor. I must say.
Okay. Um, so then you finished, you graduated or you came to the US quite some time ago? You came to Boston or
Yeah, so
where
I
did you
started
study?
I
Hmm.
started college at Anna University. I did my BTech in biotechnology there, and my early scientific influences were from there. Dr. Gautam Pennathur, shout out to him. He is a phenomenal chemist that was working on biological problems. He still is, and one of the sharpest minds I think that I've ever come across. But he helped hone my interest in uh, the chemistry and physics of biology. Let's put it that way. And then I
Okay.
applied to the biophysics PhD program at Ohio State because these were the concepts I was thinking of, and I ended up getting in. and got my PhD in biophysics from Ohio State in 2018. I came here in 2012, got my PhD in 2018. And then I moved on to go to Chicago to Northwestern University, where I did a 2-year postdoc. Where I'll go into these in more detail if you want, but
Yeah, I that phrase got me interested. The chemistry and physics of
of
biology.
biology,
Can you
yeah.
expand on that?
Yeah, absolutely. So biology is for living beings, right? It's like it's very fluid. We don't think about uh, almost. Let's put it this way. Commonly, chemistry and physics are thought of as static things that are almost inanimate, and biology is about all of the animate
All
life
living
on
things,
Earth,
yes.
right? But if as you keep zooming in, every organism is made of, let's take humans. We are made of tissues, and those tissues are made of cells, and those cells are made of little molecular machines, protein and RNA, and all of the information required to run the cell is stored in a molecule called DNA, right? And then how these little molecules interact with each other is completely physics and chemistry. And that the coordinated movement of these molecules, the coordinated physics and chemistry that we're still trying to figure out the nuances of, ends up becoming biology as you zoom out.
Wow.
When you average out all of this, it becomes an organism that's living, breathing, eating, sleeping, falling in love, all of that. So
Okay. That's a wonderful explanation and a understanding of somebody who, though I did my chemistry, botany, and zoology for graduation, I've forgotten it long ago. So this helped to sort of rekindle that. But I think at that point in time, I don't know if it was as advanced as molecular biology has come a long way in the last few decades.
absolutely.
And when you finished or you did your PhD, what was the subject of your PhD?
So I studied proteins in the ear
Okay.
that are called cadherins. They adhere two cells together. So it's hard to explain without using my fingers as props because I'm used to slides and visuals.
No,
So
you're perfectly fine using your fingers. I don't have, that's
okay.
fine.
So inside the ear,
Huh,
we have like the malleus, incus, and stapes, right? Those three bones that
okay.
are they basically are vibrated when sound hits the eardrum, and
Right.
these vibrations enter the cochlea, which
Okay.
is inside the ear, that converts this sound into an electrical signal that the brain can then recognize.
Process. Okay.
Inside the cochlea, what is responsible for this is actually a small cell called the hair cell. There's very few of them. There's only 20 to 30,000 hair cells in the ear. And they are called hair cells because they have like these three stairway like structures. They are projections on top of the cell like hair. And these things move back and forth. I'm not centered. Move
Yeah.
back and forth when sound hits them. So they move like that. And at
Okay.
the tip of them, there's a small string,
Okay.
and that's a protein complex that I studied. So when sound would perturb them, the string would pull open a little channel at this tip and cause ions to go through. So calcium and magnesium that would immediately signal to the cell to send a nerve impulse.
Okay.
So the force is converted to a chemical signal. So the force causes an opening of a pore, and the chemicals, that is magnesium and calcium, flood into the cell and cause an immediate neuronal response. Now, a lot about this was not known when I started, how these proteins looked, what were their physical properties. They actually acted like a spring. And so that spring, as it wound with tension because of hearing, the force of hearing, would unravel and then open something called an ion channel, like a gate. So it's a force gated mechanotransduction process, which is the conversion of mechanical stimulus into an electrical signal.
Okay.
Yeah, so that was the focus of my PhD. I don't know if it was too long winded or too technical, but
No, no, no. What is fascinating is how do you figure out that this process is happening? Is there a system for looking into the ear and then creating the experiment that is able to measure this?
yeah,
How do you do it?
there is. So initially, it was figured out that these proteins were involved in deafness because of sequencing. So they take people's DNA from deaf people and from normal people,
Oh,
because of these non-syndromic deafness mutations, they'll sequence both the DNA and they'll find the differences. And they found that these proteins in the ear that form that string were mutated in disease. So
wow.
then there were there's this great scientist called AJ Hudspeth that basically took the hair cells from bullfrogs. They're very big, so they're easy to look at in a microscope. He put a little glass probe on them from the side of this bundle of hair, and then he moved the glass probe, and he saw that the cell was producing impulses
Wow.
through very simple techniques, right? And so with that, he was able to do rudimentary calculations and find out that this hair bundle movement is a cause for electrical impulses that stimulate the nerves. And then we use more and more, as as technology progresses, we use finer and finer techniques like um, X-ray crystallography, which is what I studied and specialized in, where you can look at the atom level structure of these molecules, and then you simulate these in a computer as to how they would behave in solution, and you stretch them like a spring, and you find out how they would behave. But you can also do electron microscopy, where you can look at these cells in very, very high detail, because of the property of electrons being able to tunnel through things. That's a little bit of quantum mechanics, but it's hard to explain that in a simple way. But
Yep.
you can look up electron microscopy, where you can look at cells and different parts of the cells in extreme detail. And they were able to see these string-like things connecting the two hairs, and in disease models, these strings were like basically coming off and not really staying together. Yeah.
So, for someone who's investigated this in such uh, granular detail, is it bad to keep wearing headphones all the time, which the current generation does?
It's bad to blare the headphones for sure, right? So
Okay.
you don't want to listen to music at
Right.
that high of a volume, only because these hair cells don't really regenerate through your life. They
Okay.
start dying off as you age, and the more extreme stimulus that you give it, the easier they die off.
Oh,
You shouldn't really be listening to to music that loud.
that
But yeah.
loud at that time. Okay. That was just a tangential thought because you went so close into it. Uh, so, after finishing your PhD, uh, what was the career that you decided to look at, or what are the things that you were considering?
So with my whole career trajectory, I think I've only been driven by curiosity. So I saw these adhesive molecules that I was working on, right? But they're very specialized for the year. I just wanted to know, okay, cell adhesion seems so cool, cells talking to each other. Here they were talking through like this one particular protein that was like like a string, right? But then I went to a lot of conferences and looked at other people looking at really cool things of how cells talk to each other. And cells talking to each other through force, mechanotransduction, allows tissues to kind of simultaneously move, like heartbeat or your skin contracting or muscle contraction and so on, right? So I went to Dr. Kathy Green's lab for a 2-year postdoc studying cell biology, a little out of my comfort zone. And I studied cardiac contraction and the desmosome. A desmosome is another adhesive organelle that helps cells stick together and also signal essential cellular functions. And so they can basically coordinatedly make movements. They can coordinatedly, for example, make skin into the multiple layered organ that it is, because you have
Wow.
dead skin on the top, and then slowly as you go to the deeper layers of skin, you have skin that is alive and signaling different things and so on, forming a protective barrier around you. Those are the concepts that I worked on in my 2-year stint there. It just broadened my view of how cells talk to each other through molecules.
Okay.
Yeah.
And then that became your full time job, or is it something that you went into after that? How did
So
it?
that was my postdoc, right?
Okay.
And then at the end of the day, I was like, okay, I'm gaining a lot of knowledge, but how can I make this knowledge useful?
Right.
Because at the end of the day, I think studying biology should be about how we can make biology work in harmony with us, where we can, in disease states, how we can cure diseases or use our understanding of biological concepts to better humanity. And that really was the calling that made me switch from an academic setting, which is fantastic to go into the granular detail as you mentioned, and go into the industry where people are trying to um, actually leverage this biology to make new and novel therapeutics that can help mankind. Um,
Okay.
yeah,
So, what are the current areas being explored in terms of how do you improve? Where do you start? Because, like you said, if there is a connection between uh, processes that happen in the ear and then the heart and the skin, which are all connected,
yeah,
and it's like seeing something to an extremely blurred lens because you have
absolutely.
some understanding of what is happening at that particular point, but how do these actually interact and how does this whole symphony come together?
It's a
Because
very,
we
very
just don't
insightful
think of
question. It's a very
yeah,
insightful question. And that's the reason why we don't have a cure for quote unquote cancer or some of the extremely biologically complex diseases that seem to affect multiple organs, that seem orchestrated in their attack on our body. It's because of this. It's because of biological redundancy and the cooperativity between all of our organs and cells, right? Um, and we are beginning to understand uh, a little bit more about this. So, in that vein, I think the most cutting edge um, therapeutic tools that are being developed right now are gene therapy
right.
for diseases that are hereditary and carried on, and you can find where in the genome they're present, the mutations are present that cause these diseases, and technology to actually edit that out and try to have a normal genome, therefore producing the normal proteins that would allow normal biology to ensue. And in the company that I'm in right now, Ensoma, we're trying to actually make that a one-shot gene therapy. So you take one injection, and suddenly the whole different set of cogs in this huge machine start spinning, and one thing leads to another, editing cells in your bone marrow to actually make the correct type of protein. um,
Can
I
you, in layman's terms, explain how that single shot works? Is it a
Let's
kind of
actually go back to how gene
single
therapy works. So,
right?
a lot of people would have recently read about CAR T cell therapy. So that's
Okay.
a cancer therapy where basically they take your T cells, which are type of an immune cell in your body, they take them out of your body, and then they edit them, and then put them back in your body, and then they're able to recognize certain types of cancer and attack them. And there have been a lot of successful, two or three successful cases now from it. It's extremely expensive, labor intensive, time consuming, but it is a sure shot for those people that have actually undergone this therapy. And it's a big breakthrough in gene editing because it paves the way for really doing this for other types of diseases. Now, let's take an example of sickle cell disease, right? It's a single point mutation on a red blood cell that causes your red blood cells to look like half moons
Right.
and be non-functional. Now, this was biology's answer to malaria in Africa, right? And
Okay.
southern parts of India, where malaria is really, really prevalent. And having this mutation made the people less likely to get malaria, and therefore they got sickle cell disease. They're still functional, they have less capacity to hold hemoglobin and so on.
Hmm.
But it's a disease that causes a lot of complications in the long run metabolically.
Okay.
So, but it can be edited with one change to the hemoglobin gene or
Wow.
the gene locus, right? And so, let's take this as a case study for Ensoma. If we can put in agents to go and basically edit the stem cells that actually make blood cells, then from your bone marrow you will start making the precursor cells that make healthy blood cells and not sickle blood cells. And for that you get cells from the bone marrow to come into the blood, which normally happens in stem cells, in a stem cell cycle life cycle. But you can use some drugs to actually get them into the bloodstream. We put in a viral capsid, just like a vaccine, but like a dead virus essentially, that holds our DNA that we want to introduce into that cell, edit these cells and allow them to go back into the bone marrow. And in that way, we have a potential therapeutic, but your body is the big test tube where this is happening. Uh,
Wow.
that's the
I think,
technology that Ensoma is working on.
yeah, the point at which you said that you use a stem cell and intervene, but this must be horrendously complex, right? Because
It's an
to
extremely
get into,
complicated problem to solve. I agree, but it is the cutting edge right now in terms of trying to um, work biology in our favor.
yeah,
Yeah,
and it sort of emphasizes the foundation point that you made about biology moving more into chemistry and physics and understanding what exactly is involved.
yeah.
So, so when what was your you first joined the company you're currently in, or did you spend some time in another company before joining
Oh, I
Ensoma?
joined. I have had the fortune and the misfortune of spending time in more than one company, in multiple companies. Um,
Okay,
I started in the industry at a CRO, where it's a contract research organization where we just
right.
solved people's problems, and it was really great for my career because coming from the academic setting, I didn't really understand the pulse of the industry. I didn't know what was hot, what were the problems people were trying to solve, and I got a big flavor for that in the year that I was in the CRO. I got
Okay,
so many different projects from so many different clients, and I was able to look at all the cool things people are trying to solve as medical problems, right? So,
give me
but
a
then
couple of examples that you probably, without violating any
of
confidentiality,
course,
yeah, give me a couple of things that you probably.
like a lot of cancer therapeutics,
Okay,
uh, antibody based therapeutics, where antibodies will carry the drug required to kill cancer cells, and the antibodies will go and bind to cancer cell surfaces, and they're engineered to do that. So that's a enhanced way to actually deliver these drugs right to the site of cancer, and not randomly all the cells like you would get from a traditional chemotherapeutic and so on, and things like that, um,
yeah,
that I worked on. Um, and but then after a year, you start seeing the same projects over and over again, right? Because it's
okay,
a year is a short time span in the development of a drug and stuff like that. So I was like, okay, I got a pulse of what's at the industry now. I um, I then moved to a company called Kodiak Biosciences, where they worked on uh, a lot of immuno-oncology, they call it, because it's immune cells responding to cancer cells and trying to leverage that interaction. And I worked with um, some very cool scientists trying to solve this problem. Unfortunately, they ran out of funding and had to close shop. So I was back on the. I mean, it's COVID. COVID was ending.
right,
The financial environment was crazy. So then, so the company went under, even though the science was very novel and really cool. So I was back on the job market, and this opportunity at Ensoma came up, and it seemed to kind of pick up where I left off at Kodiak, where I was
okay,
working on similar problems. And now I'm here engineering antibodies and proteins on stem cells to try and figure out how to get certain proteins to be activated and certain proteins to be, um, you know, uh, to be downregulated or basically be
right,
chewed up, so that you can select for certain populations of stem cells that will be beneficial for our technology.
okay,
Yeah.
terrific. So one of the things that I'm observing, Avinash, is that we used to have silos when
Mm.
you were studying early on.
Mhm.
Chemistry was different from physics, was different from. There's
Mhm.
now a convergence of streams, of various career
Absolutely.
streams. And
Absolutely.
that is leading to new opportunities in terms of figuring out how exactly this is going to go and what are the. And I think cancer is the big prize because it's one of those things that has not been tamed for the last, so many years, 40 years.
Mhm.
And the cancer is only getting more complex in terms of given our current living conditions and the pollution that's happening and the way the bodies are now changing because of various other things which are there which never used to be there 40 years ago, microplastics and
Yeah.
various other
You
things.
hit the
And
nail on the head. Cancer, in my opinion,
hmm,
is a metabolic disease
right,
because our bodies have evolved over millions of years, but our lifestyle has changed in a matter of decades. So our body has not caught up to the lack of sunlight, not being able to spend time outdoors, not being able to eat whole foods, not being able to walk 10 to 12 kilometers a day, which is what the humans were designed to do
right,
even 1,000 years ago. 1,000 years is a blink in an eye for evolution. But in the last 30 years, we've been sitting in the dark, being affected by blue light until 2 a.m., eating Swiggy and Uber Eats at ungodly hours, and not being in touch with our circadian rhythm, eating
yeah,
processed foods that really trigger our biology in a bad way.
right.
And so, really, when we're trying to find a cure for cancer, we're actually trying to solve a problem that we created.
And
Um,
there's a delicious irony that the blue screens are being used to solve the problems themselves because all of these things are happening at a level where you can't see anything without going very deep into the body. It's not, between, it's so far below our visual threshold or even our auditory or any other threshold
mhm.
that you have to use specialized instrumentation to even get to those places.
I
So,
agree. But if it's too late, I
yeah,
agree with that.
yeah,
But but the body is miraculous.
yeah,
The amount of redundant, mechanisms that we have for recovery are
right.
actually tremendously powerful. If even if we stop consuming blue light after sunset, eat in the times that the sun is up, which is when the enzymes are available to actually digest that food, go to bed at a good hour and wake up early and look at sunlight early in the morning to reset your circadian rhythm, drink a lot of water and drink whole natural foods, uh, we wouldn't be in this mess. And we could actually revert this mess.
Yeah,
And I think another parallel future in therapeutics and understanding that is really prescribing a good diet,
right,
sleep, exercise, and complete lifestyle changes
right.
that go away from the norm that is right now, right? Because I almost feel like it's easier to stop cancer at that stage when someone can recognize their metabolic markers, go get blood tests often, look at all the metabolic markers that cause inflammation, cell disease, metabolic disease, and switch your lifestyle to actually improve those markers. You can easily go by not going to the doctor ever.
Okay.
I truly believe that. I truly believe biology is that powerful.
It
So
is. And like you said, it's almost like 1,000 years is a blink in an evolutionary timeline.
yeah,
But the amount of change that has happened, let alone 100 years, I think for my generation, I'm
last
in
40
my
years,
60s.
absolutely.
Yeah. And 30 years later, it's like we're looking at things that we would not have dreamed of
No.
when we were in school or in college. So I think life is just getting faster because we are also figuring out that we can now travel across the world, get to any part of the world that we want to in the shortest possible time. We're changing food habits more dramatically than most generations before us would have.
Mhm.
And we probably don't want to go back to a time which was simpler.
Absolutely.
That, that's one of the things which we are finding out is very hard to do. That the prescription is there, but I'm doubtful if most people want to embrace that right now.
I completely agree because the threshold for metabolic disease hasn't been met, right?
Yeah.
Once people en masse start getting sick because of the snowball of this dramatic lifestyle change, evolutionarily speaking. Then it's gonna go pretty nuts, because
Yeah.
already the mental health of people is in a huge decline, and physical health will follow,
Yeah.
unless we wake up a little bit.
Yeah.
I mean, we can try and make therapeutics for as many diseases as we possibly can, but we can never keep up with the rise of technology, and how we've forgotten to live with our own nature. And that should be with Mother Nature, but
Yeah.
also our own nature. Like, we're diurnal animals that require the sun. We're people that follow the sun, and we go to sleep at night, and we eat a certain way. We're sort of designed to eat a certain way. We decided to drink this much water and stuff like that. And blatantly and flagrantly flouting those rules will catch up with us, I think.
Yeah, but then if I look at the other side, what you're doing and what certain companies are now looking at is as wondrous, because in a sense none of this was apparent to us. We had to investigate our own selves
Hmm.
because we were growing sicker.
Hmm.
It's almost as if, the conditions, the involved a diagnosis of what is causing this in the first place, and to
Absolutely.
understand that we had to look at ourselves far more deeply.
Hmm.
So
It's
there is
very
this.
wild that it's almost like the precondition for innovation.
Yeah.
And now we have all of these sophisticated tools and methods and ideologies
Yeah.
to actually
Yeah.
investigate all of these things, and we know what makes a human being tick
Yeah.
now, much more, at least compared to 20
Right.
years ago.
Right.
And we should be grateful for that, I agree.
Yeah.
But we can also be mindful.
Absolutely. Absolutely. So we'll come back from a more philosophical plane to something more
Absolutely tangible.
more tangible, because, like, we can go on and on about how the world is
Absolutely, I
changing, but
agree.
at the end of the day we have our lifespans, which are anywhere from 60 to 90 years, or whatever it
Yeah,
is that human beings are in. And that's the strange part, that some people seem to get into their 90s. There's a lot more people getting to that who have probably followed some of these practices throughout their lives,
yeah,
and then managed to stay healthy for a large part of it.
yeah.
So, in terms of how do you see this field evolving? What are the things you're going to several conferences? You're doing work on a daily basis to try and. So, what's a daily schedule like?
Well, it goes week by week, right? Like, you
Right,
plan your experiments week by week,
right,
because they're usually multi-day experiments, right? You go in, those are you plan the previous week with meetings with your team about what types of experiments you want to do. For example, let's say I designed a few variants of this particular protein that I'm working on. I want to try and see if I express it on cells, if it gives me
okay,
a favorable outcome. And so then I'll go and make an order for the DNA for that particular protein, try to express it outside of an organism, just in cells, purify that protein, and try to introduce it in a cellular context. So we look at cell to cell interactions and see if making changes in those proteins actually gave us a favorable cell to cell response, or downstream protein marker or biological marker that we're looking at.
okay,
Then we will take that same thing and try to put it into a mouse model and see if we see favorable biology. For example, we have a healthy mouse and a disease mouse, and we'll try to use this therapeutic agent in both mice and see if the unhealthy mouse is actually getting healthy again, or if there are any adverse effects in the healthy mouse itself, and so on. So, and and then the process moves on. A few months later, once you get all of these results, you compile them all into a beautiful report, and then you go into non-human primates. You go into macaques and chimps, and they're much more like humans. So that's where most potential drugs kind of are made or broken. And finally, you go into clinical trials and clinical studies. So this is really, you asked me what a day to day is, but this is like the journey of how a drug is developed. For me, it's basically depends on which part of the drug development journey we're in. If I'm in the design phase, I'll be designing a lot of molecules on the computer. Uh, if I'm in the testing phase, then I would have got these in house and be testing them in a test tube or with cells. And then there are teams in the lab that will work on animals and so on. So different projects are always at different stages of completion, and we'll be working on those as priority comes up. So yeah.
Okay. So all of these are kind of there's a foundational level of where you're beginning this off, because I'm trying to understand, okay, you have to design experiments and then do it. But before that, what is the point at which this starts, in the sense that
So
how do
that is
you
hypothesis generation based on the disease, right? So then you have these particular sets of diseases. It's hard to explain it without breaching confidentiality of what diseases we're working on or
fair
what
enough,
targets.
fair
but
enough.
but basically you would have a few RNA targets, protein targets, some macromolecule that's in your body that is dysregulated in disease, and then you want to find a way to make it more like normal. So you have a disease protein, you want to make it a normal protein, or you have aberrant production of that protein, and you want to fix the machinery that actually enables production of that protein, and so on.
Okay,
So that's where it starts. It's based on characterization of disease, and and is there actually a way to fix this problem?
okay,
Yeah.
okay. No, that's yeah. Now that's a logical flow. I mean, to me, that's a yeah. That gives me at least an understanding of, from the outside, of where does this whole process start. Because
Yeah.
otherwise, you're saying, okay, but how do they know that this particular thing has to happen? And like you said, from the time the human genome has been mapped, which was not very long ago.
Not very long.
Yeah. So, and I remember how that particular process was hugely expensive and took a whole lot of number of years.
Hmm.
But from then on to gene editing
Hmm.
has, almost been like a kind of leap where you say, how is it that this whole thing got compressed in such a short time frame
Yeah.
and
I
it
mean,
all
we
got
just
done?
learned to walk and now we're running faster than we can ever imagine, right? That it is kind of, it's very wild to think about it, honestly. It's not,
Yeah,
it's not linear at all. It's an exponential growth in our
exactly.
understanding of biology. Absolutely.
And that's the strange route, because maybe if you look back on this 10 years from now, you'll say what we were doing then was just the starting point, because you won't see it until you get to that point.
Absolutely. And the pace of learning is also extremely fast. In
Yes.
the matter of months, concepts change, but understanding about biology changes. Every year, when you go to all these conferences that we go to, we find extremely new, innovative, exciting things that people have understood about biology that we wouldn't have even thought of a couple of years ago, almost. We
Okay.
may have hypothesized a few things, but in the time that I started my PhD to finishing my PhD, the understanding about hearing was night and day. And similarly, as I've progressed, like uh, just different processes that you could have never thought possible 5 6 years ago are suddenly becoming possibilities, and that's a pace that I didn't think uh, science would move at when I
Okay.
started this journey. But yeah, it's made me a fast learner. It's,
You have no choice, I guess.
I have no
Okay.
choice.
What's the other part? Do you think that AI is going to play a big role in whatever happens in future, or is it already playing a role?
Yes and no. It has its place. The way that AI is right now, it has its place for well characterized problems that has a big set of solutions. AI is very good at picking the right solution from a given set of solutions if it resembles another solution. But for novel idea generation, I don't think AI is there yet, because uh, it is still artificial intelligence. It's not the real thing.
Yeah. No,
Yeah,
and I think that's the point that most people miss, that, data is available only about the past, not the future.
exactly.
That'll always be the state. You can never invent data about the future because you
No,
don't know.
you can predict. You can make great predictions.
Yeah.
AI could be really cool for hypothesis generation, but it removes the fun part of science.
But
Making
okay,
wild claims is a fun part of science.
no, you're right, Avinash. Just to come at it from a different point of view, even the hypothesis generation that AI does is basically from whatever it has already aggregated the information from the past.
Yeah,
Whereas each of us comes from backgrounds that are different, our cultural coding and our studying, capacity to aggregate things is very different, as opposed to a system which then follows certain rules. Okay. While I was talking to a biologist recently, he had a different take on this whole thing. He said
okay.
AI was adaptive intelligence. Plants have adaptive intelligence, which is why they've existed for millions of years. And they're able to adapt even though we don't see brains, we don't see any other point of,
Mhm.
you know, as to how they're progressing. So at some point, I think the artificial may get into adaptive, but that will still require a whole lot of humans guiding that.
Absolutely. And I think the adaptation comes from our end, right? Like we're the ones
Yes,
adapting to
adaptive.
add to this
Yeah,
intelligence, right? Like I mean, of course it just keeps adding information from our different data banks, but who's generating that information? It's us,
absolutely.
right?
Yes.
So it really does depend on our progress.
Yeah.
So even though it is adaptive, it's not truly adaptive. It's adaptive to 1 millisecond in the past,
Right.
almost, right?
Right.
Because
Right.
yeah.
And exactly like you said, so if I have data on one particular disease, which is, let's say, on investigations with X rays or with some form of what has been there, and I have it across a population of millions, or if I have data which is already available, then finding the patterns may be easier when it's machine driven.
Absolutely.
But when you're seeing something which is completely novel,
Hmm.
and then you're trying to figure out, okay, how does this thing actually work? Then you have to apply a whole lot of things which are not evident on the surface. And it's strange how this whole thing finally works together, because I'm sure that when you come up with a hypothesis, for example, there are creative leaps that you can't explain
You cannot
at the
explain.
beginning of the entire process.
I saw a really funny quote on Instagram once. It
Okay.
says, I don't want AI to do my art and writing so I can do my laundry and dishes. I want AI to do my laundry and dishes so I can do my art and writing. Uh,
But the-
I
yeah,
think that's the only opinion that actually matters about AI, right?
yeah, very true, very true. Okay, so you've just what? You're just a decade into your career.
Yeah.
So anyone who comes in who's looking at getting into, studying further, what are the kind of things that you would want to tell them to stay focused, and what are the things that they should be picking up?
I think they should just stay creative. They
Okay.
should stay hungry to learn and they should stay creative. And follow your passions, honestly, because the best people in any field are people that truly enjoyed what they did and
Hmm.
truly enjoyed the journey that they took. It's not easy. No journey is easy. Especially the academic journey is pretty daunting and painstaking. But um, if you keep true learning in mind and true curiosity about whatever you're learning about, I think you'll be really good at what you do. Uh,
Okay, so let me take you back to that point in Anna University where you mentioned a particular teacher who kind of
yeah,
opened up your mind. What were his classes like? What did you?
he's completely wild. Dr. Gautam was completely wild. We
Okay,
would all sit on the floor, and if he really liked you, he'd make you get off the chairs and come sit on the floor in front of him.
okay,
And he will teach you about all of these different analytical methods and ways to look at molecules and their properties and almost personify these molecules and try to give them a personality, try to give them It was very engrossing sitting in his classes, and it only spurred curiosity, nothing else really.
Okay.
And so then I went to his lab, and everything was intuitive, everything was feeling, oh, what if I add this to this, what will happen? It was truly curiosity driven science, and a curiosity driven attitude, which uh, I tried to imbibe, and I try to follow even to this day. uh, and I think that's what a scientist should strive to be and should achieve to be, or at least it's the most enjoyable part of science that has stuck with me
Yeah.
this long. Yeah.
Again, we come back to the basics every time, Avinash. Some of us are lucky to have great teachers who kind of kindle our mind at that particular point.
Yeah.
And then they brought in a sense of lifelong learning, which curiosity, like you said, for example, is one of the things that keeps you going on and on. It's not something which you say that, um, okay, I'm interested in this. And once I learn about this, it's not as if it stops. You just, it's a rabbit hole with no end at all.
Absolutely. That's what makes it fun. It's kind of weird, but that's what makes it fun. It's not
Yeah.
like
So,
one of the video games that we play and the game's over. Now what?
yeah.
there is no game over here.
Yeah, that's a lovely analogy because it's literally like you don't know what is the final level, or you don't know at which level you're successful, and
Exactly.
which is the level at which you will take a leap, because none of these things. And the intuitive part, like you said, uh, a machine can give you a whole set of results. But from that, finding out intuitively which one is the better one,
Yeah. I don't think you can code intuition. Intuition
you
is,
can't. You can't.
yeah, it's a feeling inside you. However scientific my method and my training has been, I rely a lot on my intuition for successful science. And
Okay.
I know a lot of successful scientists that do rely on their intuition.
Okay.
You have ample training for the quantitative parts and the systematic parts, and there's good lab practices and good documentation. All of these things are essential to good science. But what makes science exciting is, I think, that intuition.
Great. So, what's a good day at work? And what's a
What's a
bad day at work?
ah, that's a great question. Um, a good day at work is you had a really tough problem to solve,
Yeah.
and you have a very complicated experiment
Right.
that you've been dreading setting up, because you know that there's so many points along the way that it can fail. But against all odds, at the end of the experiment, even though it was not perfect, and no science is perfect,
Right.
you learned something that helped you further that whole chain of thought, or that whole project, or that whole set of knowledge, right? You added knowledge, and you're moving that problem forward. You're progressing in trying to solve that problem, right? That's a great day at the office, right? A bad day at the office is you're trying to repeat things that you did before, and they're not repeating. The
Okay.
worst day, the worst day is
Okay.
that you're not able to reproduce science that was done before.
Okay.
Typically, it's not science that you have done before. If at least if it's science that you had done before, you can at least try and figure out what's going on. But if it's science that you have inherited and you're unable to repeat things that were done previously, then it's the worst thing, because you already have all of these preformed biases about what has gone on based on previous data, and now you
Right.
can make no sense of it. So it almost feels like it's all gone to waste.
Yeah. No, that's a great analysis of what good and bad means. And I think this applies to a number of professions, but the underlying thought is whether an idea that you're getting or whatever it is that you're working on, there's a sense of a breakthrough,
Hmm.
or you've done something that you believe will have implications for the future.
Hmm.
That's among the most satisfying bits.
uh, Even if I've learned something completely new, even if it's not a perfect experiment and perfect results, if
Yes.
I've learned something new that I didn't know before, and it's furthered my knowledge about what I was looking for,
Yeah.
I think that is
That's
extremely,
amazing. Yeah.
it's extremely fruitful.
Yeah, yeah,
Be in
yeah,
the office, you know.
yeah, yeah. So, what is the difference the between Avinash the student and Avinash the employee or somebody who's working now, who's working?
I honestly have not really veered too much from my student persona
Wonderful.
because I go into work every day trying to learn something new. Uh, and that's what uh, keeps me in science.
Yeah,
That's why I want to be a scientist, because I keep learning new things and it's so cool.
absolutely. That's wonderful, Avinash, because that's exactly what I'm trying to look at in terms of this show's context.
Hmm.
That the first job is just a starting point, and when you go ahead and then figure out what is it that you want to do,
Hmm.
every decade almost, or every like you said, the markers, the milestones may not be every 10 years, they could be every 5 years, they could be every
Hmm.
2 years, but at the end of the day, and you're not sure because there are so many things over which you don't have control. So the fact that you luck into a good teacher, the fact that you work at a place which affords you the facilities that you want to work in, uh, the success, like you said, the bad days are when you were not able to replicate something that you thought was common knowledge, and
Hmm.
then you know, but that's the good parts keep you going because
Absolutely. Absolutely.
okay. So where do you think molecular biology is going?
You actually mentioned it a lot during this podcast about
Okay.
silos and collaboration. And really, I think a lot of confluence of knowledge is happening right now. And
Okay.
so we keep inventing new fields, but there is no field, right? It's just understanding biology from all these aspects and having this collaborative way to push problems uh, out of the way and find solutions. And molecular biology is a set, there's a set of tools in molecular biology that are really important in solving a lot of uh, problems in biology, questions in biology, just like there are in the other disciplines of biology. And what I see in the future is like this big confluence of knowledge. Everyone's starting to work together much more cohesively, like than ever before, I think. And people are understanding other people's discipline uh, very quickly. And so uh, I'm not just a protein biochemist now, I'm not just a molecular biologist. I've learned so much about animal models, about drug development, about the process, regulatory processes of how to make a drug, something that goes into the clinic, about logistics, how do I transport things. Like all of this stuff starts becoming mishmashed into this whole process, and follows the purpose of the company, or the purpose of this really cool novel technology, and how to cure these crazy diseases. So yeah, I just see molecular biology merging with a lot of other fields to actually gain better understanding uh, in these difficult scenarios.
Yeah. The collaboration is going to be a really important aspect of how this whole thing moves ahead. And it's amazing how we as humans have, this thing is almost along parallel tracks that when one era ends and the next one begins, it's all doom and gloom. But the moment it moves into another phase, then you say that I remember at the start of the internet in 1995, it was supposed to be that everything is going to be wonderful because the world is going to collaborate. But
Hmm.
then you had social media, which brought about a whole lot of divisions. Today, you found that the difference between the truth and what we called a lie has blurred. We had very clear notions about good and bad, but
Hmm.
those are fusing as well. So I think, like you said, the convergence is not just about beliefs which are very finely uh, marked, but about a blurring of things where that good and bad is actually two sides of the same coin, and we begin to understand that better.
Yeah, absolutely. And it's, I wouldn't even say blurring. I would be like, in, in photography and stuff like that, they add noise to make an image crisper, right? So like it's basically adding all of these pieces together to make actually the picture much more crisp.
Yeah,
Because if you just look at something with one color,
yeah,
you're not going to see the nuances in it.
right,
But once you
right.
add a lot of these colors, then you actually see the beauty of what you're looking at. So
Yeah,
yeah.
yeah. No, that's the fun and the wonderful part of what you do. So, in irrespective of the field that you're in, it's a question of what you bring to what you're doing that actually brings a lot more into your job.
Hmm.
And I guess that's the way it will be for people who enjoy what they're doing, which is exactly where you are right now. I can see that you really enjoy what you're doing.
Absolutely. I'm having a blast. I'm looking
Yeah,
for the next thing to learn always. And
yeah,
yeah, I can't wait. That's what it is.
yeah. Great. So, thank you very much for being on the show
Thank you so
because
much for having me. It was an absolute pleasure. I love talking about all of the things that I normally nerd out about, and I was able to freely talk about it. So I'm very grateful for that.
and that's the thing that I find interesting about freewheeling conversations because it's not so much a career graph alone as much as what are the things that you learn along the way and how do you sort of understand a little more about what goes on. And for me, this has been a completely uh, amazing process every week because I'm talking to a banker one week, a person who's into project management the next, and there was program management, and somebody who does a catering job. And so it's not. So you say that there are so many
All these
shades.
excellent stories that start getting weaved in a freewheeling conversation, right?
Yeah,
Like all these beautiful stories that you're uncovering, and then it's very engaging for people to listen to because it's not structured. It's
yeah,
not, it's just where the conversation takes you,
yeah,
and
yeah.
how the story weaves itself, which is really cool. I really find it inspirational that you're doing this, and I'm looking forward to the future podcasts and listening to them as well.
And I'm saying the more people, at some point, this tapestry comes out of exactly what you're saying. Our genes might start out of just the four basic
So for
amino
basis,
acids.
yeah,
Yeah. And then it just goes in so many places that over millions of years, you still find new things happening.
yep. That's the beauty of biology. We can't figure it out, and that's why we're excited about it.
Great. Thank you very much for being on the show, Avinash, and all the very best for the future.
Thank you
Okay.
so much. All right. Bye bye.
Bye bye. Thank you. Oh, I think I just one moment. Yeah, I'll continue to talk to you a little later. Yeah,
Okay, sure.
one
Yeah.
moment. Yeah. That was Avinash Jaiganesh, and he gave us a perspective about molecular biology, which is not from the structured and known basis of what appears in textbooks, but as somebody who does the job on a daily basis. Come back next week for another podcast with another career journey, and we'll try and keep it as engaging as possible. And thank you, as always, for listening.